Hello, Field Guides fans. Bill Michaelek here. Before we get into the episode, I want to give a little bit of background because this one is a little different. 8 years ago, we released a bonus episode featuring Gordon Maupin, one of the hosts of a wonderful nature podcast called Wild Ideas. For listeners who've never heard of it, Wild Ideas was a weekly show that ran from 2009 to 2014. And each week, Gordon and his two co-hosts, Joanne Ballback, a science educator, and Gary Popotnick, a conservation biologist, each brought a seasonal topic to talk about. It was enthusiastic and very much rooted in the idea that you don't need to be an expert to be fascinated by the world around you. And honestly, when Wild Ideas ended, it was part of what inspired Steve and me to create the Field Guides. We loved how the show had been this place for natural history, curiosity, tangents, questions, and above all, enthusiasm. And when it was gone, we found ourselves missing all that in the shape of a podcast. And it was a big part of how we ended up making this show. Recently, a new listener reached out and asked where he could find the old episodes of Wild Ideas. And that led me to email Gordon, who I hadn't talked to since that bonus episode back in 2018. Gordon very graciously shared the old episode files, and that listener is now working on getting them back online so more people can enjoy them. We'll keep you updated on that. And then Gordon did something I didn't expect. He mentioned that he was going to be traveling through New York, and he offered to record another episode with us. So we got the date on the books, and we started talking about what the episode should be, and we eventually landed on the idea that rather than do a normal Field Guides episode, we'd make this one feel more like an old episode of Wild Ideas, with each of us bringing a topic to the table to discuss. The original plan was to meet at the Montezuma National Wildlife Refuge in central New York, but heavy rains, they pushed us further west, so we ended up recording at the Iroquois National Wildlife Refuge instead, and we'll talk about more of that site during the episode. So what you're about to hear, it's a little bit Field Guides, it's a little bit Wild Ideas, and it's a chance for us to reconnect with someone whose work helped inspire this show in the first place. One thing to keep in mind, though, we cover a lot of ground info-wise in this one, and we also asked a lot of questions along the way. So putting together the episode notes for this one, it required almost as much research time as prepping for the episode itself. So we're including the episode notes with this one. Stick around to the end, and I'll be sharing them. And if you were a fan of Wild Ideas, stick around to the very end for a special treat. Okay, here's our conversation with Gordon Maupin. Enjoy. Hello, and welcome to the Field Guides. I'm Bill, and I'm here with Steve. Good afternoon, Steve.
Good afternoon, Bill.
And we're also here with Gordon. Good afternoon, Gordon.
Howdy.
And
Whose phone is on?
Someone's phone is on?
I just heard a ding.
Is it mine? Yeah, it might be mine. Are we going to leave that in?
Yeah, why not? I'll just airplane mode.
Yeah.
All right, there we go.
Okay.
Did I turn mine off?
No. You're fine. I just don't think it's ever happened before. Even though I never put my phone into airplane mode or anything.
Oh, no, we get dings.
We do?
We did in the last episode, yeah. You must not have noticed.
I got it on silent.
All right.
Or I must not have listened.
And what we're going to do today is get you out into the field, in the woods, and on the trail. Each episode, we pick a natural history topic, research the science behind that topic, and then take you out to a natural spot and share with you everything that we've learned. And as we said in the intro, we have a special episode today because we are going to be mimicking what the great podcast Wild Ideas used to do on their show. Instead of just one topic, you get 3 topics for the price of 1. So I'm actually going to take a page from the Wild Ideas script, and I'm going to say, Steve, what's going on outside?
Oh, I like that.
Oh, I love it.
But before we do that,
Yeah.
I was so excited to say, Steve, what's going on outside? I forgot that we should talk about where we are right now.
Oh, yeah, that's a
Right?
good point.
So initially, we had made plans to meet at the Montezuma National Wildlife Refuge, which is farther east, more in central New York, but the weather just did not cooperate. So instead, we are here at the Iroquois National Wildlife Refuge. It's noon. So instead, we're here at the Iroquois National Wildlife Refuge, and I just want to give a little background on the site before we get into each of our topics. So this place, it is a wetland complex, and if you're familiar with Western New York, it's almost exactly between Buffalo and Rochester. And if you're a little less familiar with Western New York, we could say we're a little bit south of Lake Ontario, and we're northeast of Lake Erie. So this is an important stopover site for a lot of bird species when they're migrating.
Oh, we find ourselves here every birdathon.
Oh, right.
Yeah.
Yeah. And this site here, it's the centerpiece of what a lot of people call the Alabama swamps, which is this massive 20,000 acre wetland area.
Well, that's sort of why I said, are we in Alabama now? Because when I was looking up weather, I even checked Alabama, but that was for the one we were going to. And I was like, are the Alabama swamps, is that considered where Iroquois is? Or is that the extra 2 hours away?
No. So the town of Alabama is nearby, and that's why this is called by some people the Alabama swamps.
Yeah.
But Iroquois just takes up part of it. But it's unique because what we have here is a state and federal partnership. Because the property we're on now, this is federal land, a National Wildlife Refuge. But to the east and the west are state properties. So to the west is the Tonawanda Wildlife Management Area, and to the east is the Oak Orchard Wildlife Management Area. So together, you have almost 20,000 acres of wetlands here.
Bill, I got to interrupt.
Yeah.
There's an egret over there looking pretty great.
Oh, it's a great egret. A great egret.
Is it Arodia alba? Albus?
You're showing up.
Is that what it is?
It's a great egret.
It's a great egret.
It looks like a great blue heron, but it's white with black legs and black feet, right?
Yep.
Oh yeah, there we go. Now it just took off.
Yeah. Well, tell us more about Ottawa or
Iroquois. Yeah. So together, these properties, they form one of the largest contiguous wetlands here in the Northeast. Not the largest, but one of the largest, because Montezuma just on its own is bigger. One description I read kind of comparing the two sites said Montezuma was like an international airport. And this site here, Iroquois, the Alabama swamps, it's more like a regional airport. It's a little bit smaller. So it is an important stopover place, but it does not see the numbers that you would see at Montezuma. But we are here in mid-May, and as Steve alluded to, we were just here this past Saturday. We were doing a birdathon, which is a competition where teams in 24 hours try to see as many species as they can, and many of the teams visited Iroquois because there is such a diversity of migrants here and residents here as well. Waterfowl's the big one though, obviously because of the wetlands. So this is a pit stop along the Atlantic Flyway. As we already mentioned, it's essential habitat. Hundreds of species of migrating birds and other wildlife. I'm going to talk about one of the other migrants coming through here that's not a bird. But the habitats here, there's a mix. You're going to find marshes, you're going to find hardwood swamps, but there's also managed grasslands. And all of the water flow is managed by a system of dikes, pumps, and water control structures. And those are meant to mimic those natural flooding cycles that were lost after agriculture and other development got going here in the 1800s. So if you're looking for hard numbers, there's been 266 species of birds that have been found here. And
that was by us on Birdathon over the weekend.
I wish. And including 20 species of birds of prey.
Oh,
but
okay.
The big species, the big bird of prey that a lot of people come here to see
eagle
is the bald eagle, right?
Yeah, because you can see it from all the way across the wetland.
Yep.
It's just its white head, it's like shining, you know.
And the refuge, they do have a live eagle webcam. I was looking at it this morning. The young are fully feathered. They're looking pretty ugly, but they're making their way in the world. I don't think they've left the nest yet, but we'll link that in the episode notes so people can check that out if they want to. So that is the Iroquois National Wildlife Refuge. And Steve, now we pitch it over to you.
I'm going to try to stick with something that Wild Ideas did a lot of. So I'll admit right now, it's been a few years since I've listened to an episode of Wild Ideas. But I remember phenology being an important factor when it came to choosing topics. A lot of times it was what you guys were seeing that day or the day before or something. You tried to stay...
It's what Bill said. Our format was, what's going on outside?
That's
right.
That's
true. That's true.
So it was timely, frequently timely.
Yeah.
Right.
So for people that don't know, phenology is talking about the timing of natural events.
All right, so I chose a topic that I know almost nothing about, but I did as much reading as I could about it last night. And so for my portion of the episode, I wanted to talk about the Jefferson salamander, Ambystoma jeffersonianum. So we've actually recorded an episode about the spotted salamander emergence, and that one is Ambystoma. Have you heard that?
Yes.
I say Ambystoma. I don't actually think it rolls off the tongue in the right way. So I think it actually probably is Ambystoma.
I like Ambystoma.
I know. I just realized that I probably have been saying it wrong. But either way. So the spotted salamander is Ambystoma maculatum, and we probably mentioned the Jefferson salamander in that episode, but I think we probably barely said anything about it, so I'm just going to mention it again here.
Oh, yeah.
Yeah.
We didn't talk about it too much.
So first of all, I like to say what they look like first, so people can have an image in their head for the whole time. They're dark brown or dark gray, and they have a wide snout and long fingers.
That's where their genus name comes from.
It does.
Yep.
Do you want to?
Doesn't it mean snub-nosed or
Yeah,
blunt-nosed
ambulus means blunt and stoma means mouth.
Yeah.
So they're anywhere from about 4.5 to 8.25 inches long, and they're long and slender, and their tail is nearly half of their total length. So my favorite aspect of this species, at least in terms of the way they look, is this striking bluish flecks that are scattered along their limbs and the lower sides of the body.
Yeah, like their flanks, right?
Yeah,
Yeah.
They're just kind of like speckled blue on the sides. So Bill also already talked about the etymology, where the genus comes from, but then when we talk about the specific epithet, Jeffersonianum, I heard that it was a patronym honoring Thomas Jefferson.
Yeah, that's what I thought.
Right, but then I went to the Illinois Natural History Survey herpetology collection page, and they had their own thoughts. They said that Jeffersonianum is dedicated to Jefferson College, but
That page is known for perpetuating conspiracy theories.
I know, but the college is named for Thomas Jefferson, so
Oh.
maybe it's all
So it's an indirect.
ultimately the same thing.
Right.
So the genus Ambystoma has about 32 species across North America. That's kind of cool. I didn't realize how many species
Wow,
there
yeah.
were. And in terms of their life cycle, they have an aquatic larval state that looks like an axolotl,
Yes.
Um,
With the gills on the head.
yeah. But the adult is terrestrial and only comes back to the water for mating, and that happens sort of around this time of the year.
Yeah. Now that you're mentioning that, you said the axolotl is kind of a joke, but I think it's in the same family.
I think they're definitely closely related.
Because they have that same kind of like
They do.
smiley look
Like
because
a
of
blunt
their
nose.
blunt nose.
Yeah.
Do you know, Gordon?
No, I don't.
Yeah, so we'll have to put that in the episode notes.
Mm-hmm.
Okay. I'll bet you a beer. I'm right.
Well, you were talking about the leaf litter, but I guess specifically Jeffersons really like this like maple beech hardwood because of the leaf litter. They also don't like too acidic soils, things like that. So obviously, maple beech isn't going to result in very acidic soils, and they can be under them,
And we
keep
are in
soil moisture and everything like that.
We are in a maple beech forest right now, so there's
Yeah.
probably Jeffersons not too far away.
Oh, and I meant to mention, also fish-free, typically vernal pools. So,
Those are kind of important.
Yeah.
They do have a lot of vernal pools here at Iroquois. It's kind of one of the things they're known for.
Yeah. So
Do we need to say what vernal pools are?
go for it.
These are just these temporary, small pools that develop usually in woods that amphibians and other species rely on because there's these seasonal pools that dry up, preventing any fish from developing in them. So their larvae, their eggs, can develop safely.
Yeah, and I'm sure that drying up and the shrinking of the pools is probably already happening, or will be happening soon.
Right?
All right, so I think the coolest aspect of this species, which actually is probably the main reason I wanted to even bring it up, was because the Jefferson salamander plays a role in this species complex with 4 other members of the Ambystoma genus that exist in unisexual, all-female populations.
4
Do
other
you-
members?
Do you guys know about this?
Well, yeah, I know a little because I didn't want to reveal anything too quickly. But when I said there may be Jeffersons around,
Yeah.
I was going to say, and if we found any, there's no way we could know for sure that it was Jeffersons.
Oh, right. Yeah, I mean, when it comes to this unisexual, all-female population, they kind of break all the rules about what a species is, in my opinion. Not just my opinion. A lot of people get very confused about what's going on.
So you're going
Yeah,
to
I'll describe exactly why, if that's what you're worried about.
No, because I was just going to say the only other species that I knew was in the Jefferson complex was the blue spotted,
Oh, yeah.
which is Ambystoma laterale.
Well, that one's super important to this unisexual population because I'll tell you why later.
But there's other species within this complex, you're
Yep.
saying? Okay.
I'll just say what all of them are right now.
Okay.
So, first of all, Jeffies, which I guess
Nice.
I think I watched a Canadian news broadcast about the Jefferson salamander because they actually close off certain like highways and stuff to allow for the migration.
Yeah.
So one of the reporters did say, or known as Jeffies, you know.
I've never heard that,
Yeah,
but I love it.
I haven't either, but I kind of want to start calling them Jeffies. So it's Jeffies. You also have the blue-spotted salamander, which is the one that you mentioned, but also the smallmouth salamander, Ambystoma texanum, the eastern tiger salamander, Ambystoma tigrinum, and the streamside salamander, Ambystoma barburi. Not barburii, barburi.
Okay.
Yeah. So one of the cool things, I think people have already listened to a number of our more recent episodes, have heard me talk about polyploidy.
Oh boy.
No, well, I'm not gonna go into a deep discussion about
Say that again.
It's called polyploidy.
Say that again. Say that again. Say that again.
Very nice.
I like that. Yeah. So, as Gordon was sort of alluding to, polyploidy. So, humans are what's called diploid. We have 2 copies of our genome inside every single cell of our body. But when these, you have these unisexual all-female populations of the Ambystoma salamanders, and they can be diploid, just like humans, the 2 copies of their genome. But they can also be triploid, which is 3 copies, tetraploid, which is 4 copies, and then they can even be pentaploid, having 5 copies of a genome inside of every cell of their body.
Is that common? I've never heard
it's
of that.
less common. Triploid is like the polyploidy level of the unisex species.
Okay.
Yep.
Well, I got a puzzle. Take my evolutionary biologist stuff. When you talk about a unisex species, it makes me think of the Red Queen, which is from Alice and Through the Looking Glass. She's running as fast as she can to catch up, which is an explanation for sex. Mm-hmm. You mix these things so that species will have the ability to adapt.
Yeah.
Species adapt, not individuals adapt, but
right.
Species adapt by genetic diversity. And when you don't have sex going on, how do you do it? And maybe polyploidy is a method.
It is, and I'm actually going to say exactly what. That's an amazing thing to bring up because it's all going to come together.
So, I got to circle back to what you just said. I had no idea the Red Queen in Through the Looking Glass. That was a metaphor for sex.
Not when Lewis Carroll wrote it.
Oh, okay.
Biologists adapting that.
Gotcha. Okay.
Running as fast as you can and staying in the same place as a species. Trying to adjust to disease and anything needs to have genetic diversity in there.
Yeah, because I'm thinking, wow, Lewis Carroll was writing about biology. That seems a little early to be doing.
No.
So you have to imagine that when you have a unisexual all-female population, we have this idea of, have we talked about this before, Mueller's ratchet?
It sounds familiar.
Where when you just have a species that it just keeps cloning over and over again, like Gordon was saying, sexual reproduction is adaptive. There's recombination, there's mixing and matching of alleles. Like, it's great for adaptation. But when you're an asexual species, essentially just cloning, just having the same genetic information as your parents, you're pretty much only gonna be building up mutations, and those are typically bad over time. But there actually is an adaptive force in this Ambystoma complex, which it's unlike anything else in the entire world. I think this is a unique strategy, or unique reproduction strategy. So all of these unisex populations, they're derived from a hybridization event that happened 2.5 to 4 million years ago. So they've been perfectly fine for the last 4 million or so years.
They've been doing okay.
Yeah, and it actually makes them one of the longest lived vertebrate unisexual lineages. And they persist to this day due to their reproductive strategy called kleptogenesis. So what they'll do is, as we've probably talked about with salamanders before, male salamanders leave sperm all over the place. They're just these nasty little
sperm
creatures.
packets.
Yeah, spermatophores. They're just leaving their sperm everywhere. Well, that sperm is meant for members of their own species, right? Obviously, to reproduce with. But what one of these all-female lineages will do, they'll go around and they'll steal the sperm and they'll take it up into their cloaca, and the sperm triggers egg formation, but typically the sperm doesn't contribute any genetic material to the offspring. But it can happen which ties into the different polyploid levels, but also something called genome replacement that I'll talk about next. So most Ambystoma unisexuals have at least one blue-spotted salamander nuclear genome, while the other types of donated genomes can replace each other. So there are stream-side salamanders that they've actually seen replacing the genomes of Jefferson salamanders. So the unisexual individual will grab sperm from a stream side, and when they take that in, their offspring, instead of having the Jefferson genomes, do you know they typically stay the same? The mother and the offspring are typically the same. And let's say it's 2 Jeffersons and 1 blue spotted. The offspring will be a blue spotted and 2 stream sides.
So once in a while, things are getting mixed up.
Yes.
Yeah.
So and this is something that was brought up from another source that I saw. Now, this switching out of different genomes. Remember, the blue spotted pretty much is there all the time. The other ones are the ones that'll swap out sometimes. Imagine if you're existing in a pond with stream-side salamanders, but your genome only has Jefferson genomes in it, Jefferson and blue spotted. Now, you probably, when you think about like an adaptation, you might be like, well, these Jeffersons are really well adapted to this specific pond. And it turns out that when the unisex individuals coexist in the same pond with this other species, replacement is more likely than another species like from another pond.
Okay.
Isn't that kind of weird though?
That is strange.
Like, how does it know? You know what I mean? Like, how does it know to take in the thing that's like more adapted towards that environment?
Is it just chance that it happens?
Well, there's compatibility issues with that kind of gene mixing.
Yeah.
So,
but here's the thing. I think that all compatibility is a problem across the board, because I think when it comes to genetic differentiation, a lot of these Ambystomas are like just as genetically diverse as like humans are with like gorillas. It would be like if we did Bill's genome and it turned out he had a genome from his mother, genome from his father, and a genome from a gorilla.
are you sure he doesn't?
but then he's still a normal functioning Bill human. You know what I mean?
Right.
It's sort of a really weird thing, is that you have these highly divergent genomes that are coming together in this really weird unisexual population species complex.
But it's been working for him for millions of years.
But it's been working for millions and millions of years.
Well, for a species to persist, you've got to have this genetic diversity somehow, which is why I was asking about how do they last that long with no sex?
Right.
And it's because they just keep swapping out genomes
Once in a while.
Yeah, it happens at a particular rate, and I should say that when the female does lay eggs, a lot of those eggs don't develop properly, and they never become young. So it's like, I think it's most of them don't become young, and only a few of them end up surviving.
When this genome swapping happens.
No, just in general.
Oh, okay.
Yeah.
Well,
And actually,
it's amazing to me to hear this. You're talking about animals doing this, because I think of plants as being, polyploidy being not that uncommon.
Oh yeah, plants, it's everywhere.
It's everywhere. But to hear it in animals, I think, whoa.
I know, it's wild. It's like, typically polyploidy is bad, which is why we don't see it very often. But for some reason, it works beautifully for these guys. Yeah. So one thing that I think is sort of interesting, though, is that the mitochondrial genome of the all-female populations are most similar to stream-side salamanders, suggesting that this population probably originated from a stream-side-like ancestor. However, the stream-side salamander is the least common sperm donor. Actually, it wasn't even discovered to be a sperm donor until about 2009. So
recently.
But there is something else that's kind of interesting about the stream-side. When it does do sperm donation, its sperm does trigger egg development, but it doesn't replace existing genomes. So instead, it just adds an extra set, always raising the ploidy level of the offspring to either tetraploid or pentaploid, instead of being triploid, which is the typical ploidy level of this group. This also probably explains why detecting nuclear genomes that include stream sides are rare, because they only contribute genomes and don't replace the genomes from other species. So given enough time, you can imagine that the stream side genomes would eventually be replaced by other species genomes when their sperm is what's used for egg development.
Yeah. Now, do you know when you have, say, a blue spotted and it has streamside salamander genome
Yeah.
mixed in. Does that change its appearance at all?
Yeah, so the guy who actually discovered this, I can't remember what his name is, but it was in the 1930s, and he was following what he thought were Jefferson salamanders. But he started noticing that there was these different populations that were looking a little bit different. Maybe they were a little bigger. He called one like a light morph. But also he started noticing that way more of their eggs were also not developing. And he also noticed that these were all female populations as well. So he ended up coming up with this hypothesis that these salamanders were eventually just going to become extinct, because slowly they were all becoming female and most of their eggs were dying, and they were removing males from the population. Obviously, in the 30s, we had no idea about this whole complex and this weird asexual population that's been around for millions of years. But it is sort of like a kind of a crazy thought. It's like, why is it happening that all the males are disappearing from the population? Obviously, the females are, they look a little bit different, and most of their eggs are dying. Like, what's going on? But, Well,
because the reason I'm asking is because I know about Jeffersons and blue spotteds because we have them in this area.
Mm-hmm.
But I know, and I've been told many times, that you cannot ID them if you find one,
Mm-hmm.
because they hybridize so readily.
Yeah.
And then you get these intermediate appearances to them. And the only way you can really figure out which is through genetic testing.
To be honest,
No.
I didn't look at any descriptions that said this is what to look for the unisex population. This is what to look for Jeffersons and so on.
Yeah, like if around here, if you try to put into iNaturalist, oh, this is a Jefferson salamander, someone's going to be like, no. All you can put is the unisexual complex.
Uh-huh.
So my question is, and I'll look into this and put it in the episode notes, is these other species that you mentioned, as far as I know, they're not around here. So that begs two questions. One, in these areas where all the species overlap,
Mm-hmm.
does their appearance change? Or are the other species that you mentioned, besides the Jefferson and the blue spotted, the two that I know form intermediate,
Yeah.
do those species still look like they usually do?
I wish I would have.
Yeah, and that's okay.
Would have
So
made notes
that's
on it,
something
but-
we'll look up. Do you know, Gordon? Not sure? Yeah. Because as far we don't have tiger salamanders around here.
Mm-hmm.
We don't have streamside salamanders. What was the other one? There was something smallmouth?
Yeah.
Yeah, that one doesn't sound familiar.
Yeah. So it's the Jefferson, blue spotted, smallmouth, eastern tiger, and streamside.
Okay.
And speaking of all 5 of those species, they kind of crunched the numbers on it, and as of 2019, there have been 24 different nuclear genomic combinations identified. So obviously there's a lot of mixing and matching. So they found 25 unique combinations so far.
Just from those 5.
Or 24 combinations so far, yeah. All right. So who should I ask next?
All right. Gordon, what's going on outside?
Well, when I was going to come here, I was thinking, well, what should I talk about? Oh, we're going to go to a wetland. Oh, maybe I'll talk about cattails. And, that would be a great subject, except I was visiting family, and I happened to walk out, and they had a little pond behind their place, and it was covered with duckweed.
Nice.
And I got to looking at the duckweed, and I thought, oh my, that is a cool topic. Mm-hmm. Duckweed, if you go and you find, and it's usually a pretty, mostly permanent pool of water, like a pond or something, very occasionally flowing, but only very, very slowly. And as the season progresses, it will gradually get covered by these light green things floating on top of the water. They're little discs.
Tiny.
Tiny.
Yeah.
And people often will, I've had people many times say to me, oh, look at all the algae on that pond.
Right.
And it ain't algae.
Mm-hmm.
The duckweed is like astounding. It's a flowering plant, and here in the last couple of years I'd only ever seen it flowering one time, and that was because a friend found it flowering and happened to notice it and put some in a little jar and brought it in and showed it to me.
Beautiful.
And you see these little tiny discs just a few millimeters long, and oblong, and sometimes you'll see another one budding off of it, but every once in a while that disc will split open, and two little anthers and a stamen will come out, and it's a flowering plant, and they produce seed, and the seed sinks to the bottom, but mostly it reproduces vegetatively, and only for conditions that, as far as I know, remain mysterious.
that was the budding off you were talking about.
They bud off vegetatively, but only under mysterious conditions that I don't know about do they decide I'm going to go ahead and flower and start sexually reproducing.
Steve, have you ever seen a flower?
You have to look close.
I never have.
You would walk by a million of them, and
Sure.
they might be in flower, and you never notice because you got to look at them with a hand lens, pretty much.
So I'll be honest, I think I have, but it's because I'm trying to think, there's two species that I think, when I think of duckweed, I think of like the lemna, which is
yeah.
the kind of the longer ones, right?
Yeah, well, and there's a lot of species of lemna, so then it gets confusing.
But then there's another one that's like, it ends in like mycorrhizae.
Wolfia.
What is it?
Wolfia?
Man, I don't think it was Wolfia. But are they in the same genus as Amorphophallus?
I don't know.
The Arum family? Do you know?
Well, I was going to talk about that.
Okay, because I was going to say, my lab was working with a genome at the time of Amorphophallus.
Okay.
And then sort of for fun, while we were out on a kind of a lab hike, we had seen some, another member of the same family, and we're like, we're just dealing with Amorphophallus, and maybe, I don't even know if that'll come up, but we decided to collect some and look at it under a microscope and stuff, and I think we might have seen a flower, but...
Okay.
Where were we?
Oh, sorry. We're just talking about the flowering.
You were talking about how under mysterious conditions it decides to flower.
Yeah, so it flowers. So, you're a naturalist and you've never paid much attention to duckweed. You see, it's duckweed, okay, go on to the next thing. But then when you learn that this little thing is a flowering plant, you sort of get curious. What in the world? It's not only a flowering plant, it used to be a vascular plant. And there are remnants in these little discs of vascular tissue, and they are still somewhat functional, moving thing around.
Whoa.
They have stomata, or maybe one stomate on top of one of these discs. And so they're plants that actually occupied the land, and somehow they evolved to return to the water. So you think about this, what in the, and it turns out everybody knows Jack in the Pulpit, everybody knows
Skunk cabbage?
Thank you. The age, it gets to you. Everybody knows skunk cabbage around here, or if you're lucky, you get to find a green dragon now and then.
Right.
Yeah.
And this is the arum family. Mm-hmm. And apparently, the taxonomists, the lumpers and the splitters argue back and forth, should the duckweed still be in the arum family, or do they deserve to be in their own family? I kind of lean towards that. They're so different, they ought to be in their own family. But some genetic biologist will yell at me for that.
Right.
But anyway, they evolved to go back into the water, and evolution, if it doesn't need it, it goes away. Because it takes energy to make things you don't need.
Right.
And these one line of these plants started getting wetter and wetter, and eventually was doing really, really well, enough to create an ecological niche where it could proliferate and speciate, because there's quite a few different species. Now, the species I've seen most is Lemna minor, which is a North American and widespread. And once you look at duckweed, you kind of don't really go down and try to identify it, because I seldom carry a microscope in my backpack. And so I can't tell the different species apart just by looking at them. No doubt there are people that can. But you look at them, they even have the lemnas especially have little filamentous roots that don't have root hairs on them because they don't need them.
They kind of look like the root hairs a little bit.
They look, the whole root looks like a root hair.
Yeah.
It's a little kind of transparent white thing that dangles down in the water. And so this is a flowering plant that has a lot of the genetic apparatus that trees have.
Mm-hmm.
and it just floats on the water. And so I thought at a wetland, as we walk along here, I'll look left and right at a slow-moving water, and I'm gonna see duckweed everywhere. But we're not.
We're not.
We could keep walking and try to find some.
But what is interesting about this is that duckweed is getting a lot of interest in science nowadays, because it floats on the water, and if the water is loaded with nutrients, it sucks those nutrients out. It pulls out the nitrogen and the phosphorus and improves water quality quite a bit. And so it's gotten to be a lot of interest in that. And they call it gets the name duckweed because ducks and lots of other things, fish and all waterfowl and everything eats the stuff, because again, a higher plant, not just an algae, it's high in protein. It's very high in protein. And looking up on it, NASA is actually looking at, on a flight to Mars, needing vegetation and protein, that you can put a nutrient puddle in the spacecraft and grow duckweed on it, and the astronauts could eat it, and people can eat it. And in some cultures, they do harvest duckweed and eat it as a green vegetable high in protein. So it's a pretty cool plant. And when I would urge your listeners, when they see a pond, a very slow moving, I've seen duckweed in streams, flowing streams, and you'll see it along the edge and some backwater eddy things occasionally, but that's pretty rare.
Yeah.
It's often just in ponds where the water's not traveling, or a wetland where the water is moving very, very slowly, so it has time to proliferate on the surface. And it's just really a cool plant to think about.
So you're saying when our listeners see it, they can kind of go bobbing for apples, but they bob for duckweed. Well, because it's edible. A lot of cultures do it.
You could, but the fact that it likes nutrient loaded stuff, I might want to rinse it off in a little Clorox water or something.
Sure.
Which actually brings up another little interesting topic on it, is because people have investigated this stuff because it's potentially commercial and maybe a little bit commercial, and it has uses in maybe cleaning up polluted water, nutrient polluted water. And they're on top of these little discs, and on the bottom of these little discs are little biomes of their own, of bacteria and algae and everything else. And some of these, it's nature. Some of these bacterial biomes on the duckweed cooperate with the duckweed, because cooperation is very powerful in evolutionary biology. But some of it is parasitic as well, and it is the opposite of cooperating, is detrimental. And so there's these biofilms on the duckweed itself that people have been investigating a little bit. Wow. So it's a very fascinating plant, but you see these little tiny discs floating in a pond and proliferating to where sometimes it covers the whole pond.
Yeah.
And but if you do have a pond that gets covered in that, here's the thing to do, is you skim the duckweed off the top and use it as green mulch, because it will be full of those nutrients it pulled out of the water, and those nutrients can be used if you use it as a green mulch.
Oh wow, very cool.
And some places they use it as animal feed as well,
sure,
because it's high in protein and everything. So this duckweed you see, and you think you have this pond that's covered with this algae that's kind of lighter green really than most algae.
Yeah. It's not stringy.
No, it's not stringy, and it's lighter green. But from a distance you might think you've got an algae pollution,
right,
or cyanobacteria thing going on. But it's the duckweed, and it's just I would just encourage people, just take a moment and look at that stuff.
Yes.
Sit down and get some of it in your hand. And the stuff is there are duckweed species cosmopolitan all around the world. And when you think about the mysteries of plant dispersal, we know a little bit about it, but things move like we never know. Duckweed, it's kind of easy to explain. It gets on the feet and in the feathers and everything of water birds, and they can move from place to place and carry it. And they've looked at it. Duckweed can survive for not really long periods, but long enough to fly from one pond to another.
Sure.
And on birds and or even muskrats or something, you know. And, you know, it'll if you stick your hand in there and come out in duckweed, it's going to be covered with duckweed.
I was going to say that because I've done that before. You stick it in there and it sticks to your hand.
It sticks to your hand.
Yeah.
Oh, your boots, everything.
Yeah. And so, you know, that's got to be a plant dispersal mechanism that it uses. And the stuff overwinters. If it does make seed, or it makes these little bulbed kind of things that sink to the bottom, and then you know, it can overwinter that way and then reemerge in the spring. Although some species have actually been found, they've been able to revive it when it was frozen in ice.
Whoa.
Wow.
So you know, adaptability, you know, evolution is a powerful engine for adaptability.
Right.
And so duckweed, it's easy to walk by.
It is.
And even easy just to think of it as a problem, but it's just a very cool plant.
No, I've had someone tell me it's the smallest flowering plant in the world. Do you know if that's true?
Yeah, well there are bigger duckweeds and littler duckweeds, and and there's some really little duckweeds.
Yeah. So maybe that's true. Yeah.
I almost brought that up earlier because I think Amorphophallus is the biggest flower, and I think Lemna, or one of the species, is the smallest.
There's Wolfia, and an Asian Wolfia is the smallest, and I've never seen it, but it's
still all
within
really
tiny.
It's all in the arum?
But it's all in the Araceae, or the arum family, right?
I'm arguing. I'm not going to let it be in that family.
How dare you, Gordon, take this away from the arum family?
Yeah.
That's a huge claim to fame.
Yeah, well, no, it deserves its own family.
Sure.
So you guys answer the hate mail on that.
Yeah.
I don't think we'll get too much.
See, I don't know. I'm not a huge... it doesn't bother me when like morphology or niches are so different within a family, because just look at the Ranunculaceae.
Sure.
Those guys, I know they're basal, and you know, but the diversity in that family, the buttercup family, is crazy.
Yes,
you know,
and won't argue with that.
And I pretty much, and I more or less accept that they're all the one family. But you know, I would love to have a splitter. What is it? What are they? Lumper splitter.
Well,
I would love to have like a representative from each side on the podcast and just see them go to town. Yeah.
Well, what I would say is that even among the guys that do the DNA sequencing, they're arguing.
Sure, I believe that. Yeah.
So that means I'm right.
Someone just told me if an argument lasts for more than 5 minutes, both people are wrong.
Yeah. One other thing that I wonder about Lemna, or any of these really small guys, I wonder their genome size, because I was thinking about it for even the Jefferson salamander, because their haploid genome size is like 28 gigs. So a human genome haploid is 3 gigs. The Jefferson salamander, 28 gigs. Wow. And that's just the haploid. So we were saying some are pentaploid. So imagine 28 times 5, you know, what is it, 140 gigs or whatever? Like that's... intense. So and I think both the corpse flower, the one I was trying to say before. Yeah. Have I been saying the genus wrong the whole time?
The corpse flower, that's an arum too, isn't it?
Yeah. So I think the corpse flower is the corpse flower is the biggest flower, I think. And then
the lemna.
The lemna is the smallest. So
yeah.
I think I was saying Ambystoma or something, or what was I saying?
No, you're saying Amorphophallus.
Amorphophallus. Yeah. So I think I am right.
Yeah.
Okay. The wires just got crossed. I want to make sure I wasn't saying the wrong thing this whole time. That one, I think they might both have pretty big genomes, if I'm remembering right. So I wonder, even these little guys have gigantic genomes. I think they might, but we'll put it in the notes.
You can put that.
Well, if someone's looking for a PhD topic, there you go.
We're full of those on our podcast.
Oh yeah,
we mentioned it several times.
There's
genome
still
size
so
evolution
much to be known.
Yeah.
I will say genome size evolution is a really cool topic, because then you can get into stuff like like bladderworts, those carnivorous plants, their genomes are all tiny. But of course, they don't have roots, and they don't need a number of genes for things. So there's a lot of cool topics in genome size. So
if I would be a betting man, I would bet that the duckweeds have pared down their genome size. But you never know.
It's true. I mean,
you never know.
I would say like bladderworts, they kind of have these weirdo, like they're missing a lot of flower parts, and they're missing a lot of plant parts. It seems like so maybe they have whittled it down. Worth looking into.
But sometimes you'll have the whole genome the whole gene for something, but it's inactivated.
True.
And it'll persist for thousands and thousands of years.
Right.
Yeah, it just stays tightly wound up in the genome and never really opens up to become expressed or anything.
Yeah.
So, yeah, it happens.
All right, do I get to go now?
Fine.
All right. Should we let him?
Do we want to walk a little bit? It looks
Sure.
so quiet over there.
Yes.
The wind's been fine, but every now and then it picks up.
All right. So the topic I picked today was one that
We're out of time, dude.
I do feel like it.
I hate to say it.
The topic I picked was dragonfly migration.
Oh, okay.
So this is something I've heard of, but never really dove into. And after doing this, I regret that I didn't pick this for a complete episode topic. Because like with you, it was so hard to pare this down. And there's so many points where I'm going to be like, but I can't get into that because that's a whole world unto itself. But I just want to back up to talk about how, the idea of dragonfly migration, the idea's been around for centuries, but it's only until really the last 20 years that we figured out what exactly is going on. Prior to that, most scientists, most observers, they just assumed that when they would see dragonfly swarms, these seasonal swarms, that usually dragonflies were just being pushed around by the wind. And
they don't really have much agency on their own.
No, totally. That they were just kind of gliding around. And
was anyone saying like birds, they just dive under the water for the winter?
Probably at one time.
Actually, it being an insect,
Or hummingbirds getting on the backs of geese.
Well, technically, a lot of dragonflies do spend the winter underwater.
Do they? Well,
just not in adult form.
Yeah, right. Of course.
But hummingbirds do not migrate on the backs of geese.
No, they do not.
I'll believe it when I don't see it.
And they do have legs too. So
yes.
I've had people ask if
dragonflies?
Hummingbirds have legs.
Oh, hummingbirds. Yes.
So generally it wasn't believed that dragonflies were intentionally migrating. But that began to change in the 1950s. There was a British entomologist. His name was Philip Corbett. And he had this idea that maybe some species were migrating, so he started gathering observations from all over the world. And he did start to see patterns that they appeared in certain places at certain seasons, and most importantly, they seemed to move when you piled up these observations and looked at them as a whole. In many places, they were moving consistently in one direction over long distances. So he continued working on this. Subsequent work he did in the 60s, and then he did a major synthesis in the 90s.
What do? They put little bands on their legs?
Oh, don't worry. You're not going to believe what they do.
I've heard what they do with butterflies, so I wonder if it's a similar...
Oh, yeah. No, definitely. Very similar. So he argued that some dragonfly species should be considered active migrants, like birds, and not passive drifters. But it wasn't until the 2000s, and especially into the 2010s, that more modern tracking tools finally proved him right. Species like the common green darner, that we're really going to focus on today, they do engage in organized, multi-generational migrations across North America, like the monarch butterfly. And I want to point out, this is not like insect dispersal. Some people may have heard about when insects, sometimes they'll disperse randomly to new sites to look for food or to look for new habitat. That's different. What we're talking about today is directional, it's seasonal, and it's synchronized.
Okay.
But I should also say that based on what we know now, that dragonfly migration is relatively rare within the dragonfly order, Steve. Come on.
Don't tell me.
Odonates.
What is it? Oh, yeah, odonate. Yeah,
Yep.
Odonata.
Odonata. Yeah. So there's between 5,000 and 6,000 known species. Only about 25 to 50 species are known as regular migrants. That's less than 1%.
Yeah.
Okay? But here in North America, for whatever reason, of the species that we have in North America, about 10% are migrants. So when people talk about migratory dragonflies, they're usually talking about a handful of species within the group known as the skimmers. Do you know the name for that family? Anybody?
I don't know it.
No.
Libelulidae.
I never.
It was right on the tip of my tongue.
It's fun to say.
I wonder if Jason Dombrowski was listening. He knew it, and he said it.
He's shouting it into into his car. Or the darners, and that's the Aeschynidae.
Oh, I knew that one. I wish you would have asked that one. Yeah.
So the common green darner, that's the heavy hitter of that second group, the darners, and that's what we're going to talk about. So this is Anax junius, and if listeners don't know what the common green darner is, like just think of a dragonfly. That's the common green darner, right?
It's the cartoon version.
Big or little?
Big, right?
Big.
Oh,
yes.
It's big.
2.5 to 3 inches long.
See, that's good because I think I was picturing like a bluet or something.
Oh, okay. So
A damselfly.
yeah, I was going to
actually.
say, you're picturing more of a damselfly.
Yeah.
So these guys are large. They're striking. They usually have a bright green thorax. Males have a long, slender blue abdomen. So they have like a two-tone look when they're flying. So because they're large, because of this color difference, they're more readily identifiable when you see them fly by.
When I was a kid, I was a little afraid of dragonflies.
Oh,
I was
a lot of people are, right?
I'm not taking a chance.
Yeah.
Like it looks like it could hurt me.
But they can hurt you.
They can?
They cannot.
Oh, they cannot.
Females, their abdomens are brown to olive green. But both sexes, they have the huge eyes that typically appear bright green, and they have clear wings. They are capable of asynchronous flight. That means they move each of their forewings independently. So they can fly backward, they can hover, and they can even pivot 180 degrees in a fraction of a second.
Oh, hold on. If you don't know what a dragonfly looks like, think about a number of movies by Studio Ghibli. Because for some reason, Hayao Miyazaki likes to animate these like fantasy planes and stuff with like dragonfly wings.
In the Dune movie recently, they had things they called ornithopters.
Yeah.
But they were modeled on, they looked
Yes.
remind you of dragonflies.
The thopters, they are so much more dragonfly-like than what I was thinking of. But Miyazaki still does the dragonfly look for a lot of his flight.
So if you had to guess, are green darners, are they ectothermic or endothermic? The fact that I'm asking this question.
This is interesting. One of my favorite dragonfly stories was I was riding on a bicycle and I had my son in a little carrier, because he was a little bitty guy. And I was going to the local pharmacy and I looked on the bricks in front of the pharmacy. Great big dragonfly. It's winter. It's cold. And I'm thinking, oh my. So I had to go get the dragonfly on my finger. And he was cold, so I put him on my finger and I was riding home on the bicycle. And after about a mile, I felt this guy vibrate. Oh my, did he vibrate. And then finally he flew off.
That is beautiful.
Yeah.
Because I'm going to talk about that.
Yeah, that was wonderful.
I know what happened.
Okay, tell me.
So for the listeners out there, if you don't know ectothermic, that's typically cold-blooded.
Mm-hmm.
And then endothermic.
You're getting your heat from the environment.
Correct.
When you're
typically
ecto.
the sun.
Yeah.
And then we're endothermic. And typically most insects are.
I was at ecto.
Right,
Yeah.
ecto.
So when it's cold, they're sluggish. When it's warm, they're like these mosquitoes that have been at us. Sorry, I say at us. I think they like me the most.
They like you the most.
I'm also the most exposed.
You
so,
are.
yeah.
Steve wore shorts today.
I wear shorts all the time.
But if we were standing here, if we had gotten here earlier, say at 8 o'clock, and it was in the 50s, especially the low 50s, a lot of insects would not be flying around. Their muscles are literally too cold. But green darners, they can warm up with a trick, muscular shivering.
There you go.
So they rapidly contract the flight muscles in their thorax without moving their wings. So they can generate enough heat to raise their thoracic temperature to 110 degrees Fahrenheit.
This happened on my finger.
Yeah. And you were feeling the vibration.
Yeah.
Mm-hmm.
So green darners are called facultative endotherms.
Oh, that's cool.
All right.
Yeah, very cool.
So they can generate heat when they need to.
Yeah.
They have this on-demand ability. And that gives them a huge competitive edge. Because this morning, when it's cold and all these other insects have to sit still, they can go around and pick them off. Right?
Yeah.
Maybe we keep moving, because I think when we move, I have less than 100 landing on me.
Oh, come on.
Well, the picking things off, the dragonfly, you see those globe eyes on either side of the head. It's like they can see every direction at once.
That's right. And that's what I'm going to talk about next. So, because I want to give folks an idea of just the array of adaptations that these guys have to be predators, and then we'll get into the migration a little more. So there was a paper in 2015 where researchers, they used ultra high-speed cameras to study hunting dragonflies. And they found out that dragonflies don't chase their prey. They use something equivalent to missile guidance. It's called proportional navigation. So
They shoot missiles at them.
they're just too fast for you to see.
All right, we've lost all our credibility.
So their head moves independently of their body to keep the prey locked on what you were talking about, Gordon, their visual fovea. That's the area of highest faceted resolution. And they adjust their flight trajectory accordingly. So as I said, they don't chase. If they're chasing a midge, say, from behind, they calculate the midge's velocity and they aim for a future point in space.
Oh, that's cool.
And they also have a specialized dorsal fovea on the top of their head, looks upwards, which is why they almost always attack from below, so the prey is silhouetted against the sky.
That's
Now,
cool.
Just to compare this to other predators, do you guys know, like a wolf, when they attack prey, what's their success rate? Do you guys know?
It's like 1% or 2%. I mean, it's low.
It's about 10%, 10 to 15%.
And they're really good at picking out somebody that's susceptible.
Right.
Yeah.
Lions, they have about a 25% success rate. Sharks are about 50. What do you think dragonflies are?
I'd say 90.
95%.
Oh my gosh.
Isn't that crazy?
Well,
Wow.
here's, if your listeners want an exercise in futility, get a butterfly net and try to catch a dragonfly. There's no hope.
Really? I've never tried.
Well, there
That's
you
good
go.
to know.
Then now you,
An exercise in futility.
yes.
So, in some of the reading I was doing, they were talking about dragonfly migration, and they referred to it as a moving front of apex predation.
There's about a 95% chance you're
Yeah.
not going to make it if the dragonflies migrate
Has
through.
decided, right?
Yeah.
I'm going to eat you. Let's talk about how they figured the migration out. This was a great study in 2006 where researchers decided, okay, we're going to find out what these dragonflies were doing. Were they passively drifting or were they actively migrating? So they used surgical adhesive and they attached 300 milligram radio transmitters to common green darners.
Oh my God.
Radio transmitters.
Green darners typically weigh about 1 gram. So this was about 30% of their body weight.
Wow.
And then
These ones had a less than 95%
Yes.
success rate
much.
for prey.
And then they used Cessnas and ground-based trucks, both with antennas,
Whoa.
and followed the dragonflies.
Wait, what's a Cessna?
It's a small plane.
Oh, right.
So I just love this image of researchers in planes holding antennas chasing dragonflies down the Jersey Shore.
I'm picturing, have you seen the movie Fly Away Home?
Oh, yeah.
Where the plane is like a goose,
It's
right?
like a goose, yeah.
Yeah, I'm just, but the plane is, the Cessna is the dragonfly.
dragonfly.
Yeah.
So they tracked these individuals hundreds of miles, and what they found is that dragonflies, they follow a pattern similar to songbirds. They follow what's called optimal migration theory, OMT. So this is a theory that models how animals optimize their itineraries, basically. So they adjust things like flight speed, stopover duration, and fuel load, all to minimize the travel time and their energetic expenditures or how much they're risking predation. So typically, just like birds, they would migrate in bursts of several hours. With dragonflies, it was 3 to 6 hours. And then they would rest at sites like where we are right now at Iroquois for up to 5 days to fat load. And they would be consuming midges, other insects, building up their fat stores that would fuel the next leg of the journey. And if wind was conducive to where they were heading, an individual dragonfly could travel 90 to 100 miles in a day.
Holy cow.
Now, that's extreme. Typically,
Sure.
it's more like 20 to 50, something
Right,
like
right.
that. But there were ones that went 90 to 100 miles. So this behavior, it does mimic the stopover ecology that we see in migratory songbirds.
I was about to say, it's all the same strategies
Yeah,
that birds use.
Totally.
Yeah.
And there was a 2018 study that followed up on it because this 06 study, their sample size was small. They weren't looking at a ton of dragonflies. I think I read 14 dragonflies that they were following.
Oh, wow.
And this was just in one area, like I said, the Jersey Shore. So researchers in 2018, they said, we need to look at this continental wide. They used stable hydrogen isotope analysis
Whoa.
to reconstruct how dragonflies were migrating across North America. So maybe you guys know more about this. but they said hydrogen isotope ratios in rainfall vary predictably with latitude. And as dragonfly larvae grow in ponds, they consume water and insects from the pond, and these isotope signatures, these latitudinal ones, become chemically embedded in their developing wing tissue. So it's almost like a chemical stamp.
Whoa.
Like once they analyze those isotopes, they'll know what latitude it came from. Once the wings harden, this isotopic fingerprint remains fixed for life. So this study analyzed 800 wing samples from museums, and that spanned 140 years. And by reading these wings, they're all collected at different times of year, they figured out an 1,800 mile, 3-generation migration cycle.
Wow.
For green darners.
Oh my gosh.
So the way it starts, in the spring, we can say generation 1. These are born in the south, in places like Mexico and the Gulf Coast, and they migrate northward to places like where we are right now, Iroquois or places like Montezuma. These are the tattered arrivals. Like if you see a green darner right now, and its wings are kind of beat up, that's because it migrated all the way from down south.
Wow.
They arrive here and they lay eggs. Generation 2, that's born here, they migrate to the south in autumn. Okay, so their larvae develop pretty quickly. And then they migrate south in the autumn. They've never been to these places, but they migrate thousands of miles to reach the warmer latitudes. They lay eggs and they die. And then there's a winter generation born in the south. This is the only generation that doesn't migrate. So they remain in the south over the winter, they reproduce, and out of their eggs comes the new generation 1 that comes up here.
Wow.
Oh my gosh.
So 3 generations, very similar to the monarch butterfly, right?
Sort the genetics out on that.
Right?
And then explain the navigation.
Exactly. So I did want to talk about how, I alluded to it a little before, how dragonflies use the weather. So one quote I read said, dragonflies don't fight the weather, they exploit it. There was a 2019 study that used the MODIS wildlife tracking system. So these towers that can pick up animals that have transmitters on them. And they did this with dragonflies, and they showed that they selectively migrate when weather conditions are good. Very similar to birds. They often leave right after a cold front comes through, because that cold front, it gives them 2 things. It signals when it's time to leave, when the temperature drops. And then they usually then have a strong tailwind because of the change in pressure with the cold front. And as I already mentioned, under these prime conditions, sometimes an individual dragonfly could fly 90 to 100 miles in a single day.
Right.
It's crazy. But this brings up the question of how do they navigate? This is an area that's still actively being studied. We think their main tool is the sun. They have a circadian clock that marks the sun's movement across the sky, so 15 degrees per hour, and they kind of set that internal clock with the sun's position, and then they can maintain a perfectly straight heading throughout the day. During that 2006 study that I talked about, when researchers tracked dragonflies with planes, they found that the dragonflies maintained straight headings over long distances regardless of wind.
Okay.
Oh man.
And because their direction stayed constant while the sun moved, scientists, they strongly infer a solar compass at work. So more study needs to be done. But what about when it's cloudy, right? So it's hypothesized that they switch to a magnetic compass. So this hasn't been tested in dragonflies, but researchers have proved it in other migrant insects like monarch butterflies. They used flight mills. Have you guys heard about this?
No.
It's almost, think of it like a little carousel where the insect is tethered to the middle, so they fly around in circles. And they surround these with something called Helmholtz coils. So you can create a magnetic field in there, and you can change it. When they would flip the magnetic field, the insects would instantly change flight direction. So we think they do this by sensing the angle or dip of the Earth's magnetic field. If you've ever seen those diagrams of the Earth's magnetic field coming out of the equator and going into the poles, we think insects and birds can tap into that and sense the dip. And they know, am I going toward the pole or am I going towards the equator? So again, we don't know for sure that this is what dragonflies are doing, but there does seem to be indications that that's what they're doing.
There's like a proof of concept in other species.
Yes.
They just haven't tested it yet.
That's it, proof of concept. So another PhD right there.
There's no thought that they can see in the infrared?
I didn't look into that, so
Okay.
maybe.
Okay.
Yeah, we'll put that in the episode notes.
You know, insects have a lot of insects have a much wider spectrum
Oh,
of vision.
definitely. Have you read the book Immense World by Ed Young?
No.
I would highly recommend it. It talks a lot about animal senses, and the whole section on infrared.
No, I did read it.
Yes.
Yes.
Just about how, as time has gone by, we've thought, oh, only these few animals use infrared. Oh no, more use it. Oh no, more use it.
Right.
And now we're finally realizing we're kind of the only putzes walking around without infrared.
I like the idea of every time you said, oh no, they were getting more and more scared. Not like, oh no, I was wrong. No, oh no, they can all see in the infrared.
Well, they can see in the ultraviolet too. The plants use the ultraviolet on their flowers.
Right.
Yeah.
Now, what I want to wrap up with is I don't want to give people the idea that all green darners that you might see are migratory ones. There's two coexisting strategies within populations of green darners. So you have the migratory population that we talked about. So they do that 3-generation migratory pattern. But then there is also a resident population in most places within their range. You can call these the overwinterers. So they remain here year-round. Their aquatic larvae survive winter underwater in mud in diapause. So that's their dormant state.
Mm-hmm.
And then they emerge as adults in summer, typically in July and August, after the migratory ones have already emerged.
Oh,
Okay.
Do they not breed with the migrants?
So that, I'm going to talk about that.
Okay.
Hang on.
Yeah.
Resident dragonflies, sometimes you can tell just by looking at their behavior, because residents often behave as what are called perchers. They repeatedly return to the same vegetation after short feeding flights. But the migrants, they're usually what are called, they have a behavior called hawking, where they're continuously flying, patrolling long straight routes, and they rarely perch. Now, Steve, to your question.
Mm-hmm.
Because the two groups had separate emergent peaks, kind of at different times, biologists for years thought that they were reproductively isolated.
Mm-hmm.
But more modern sampling shows that while they do have separate peaks, they overlap.
Okay. So there's a little bit of gene flow, maybe?
Yes, they form a single integrated population, and that's an ecological hedge, right?
That's cool.
By dividing your population, the species is going to ensure survival across, whatever changes may happen, environmentally, climatically, or habitat-wise.
I just wonder how the one population's chosen and the other one's chosen. If they're mixing all the time.
Right.
Or...
Yeah. How do individuals,
Do any...
quote unquote, decide?
Yeah, like the offspring, which way do they go?
Yeah. I'll look into that to see if anyone's
Interesting.
looked into it.
Well, some sort of selection event could separate those species.
Individuals, you mean?
No.
The species, right.
Separate the species into two species if there was some event that dramatically preferred one over the other.
Yeah. Or is more successful, right? Yeah.
Or is more successful, it takes longer then.
Yeah. Now, the thing that I'll leave you with is just to give people the idea of the scope of dragonfly migration. And remember, folks, there's only, a handful of species doing this. Most dragonflies are not. But just to give you an idea, there was another study in 2016 where researchers used vertical-looking radar and high-altitude balloon nets over the course of about 10 years to measure the amount of dragonflies that were migrating. They found that migrating insect biomass exceeds bird migration biomass by 8 to 1.
That's crazy.
That is crazy.
Isn't that nuts?
Can you imagine what it was like before all the wetlands were drained?
Right.
Oh
Right.
my gosh.
And that was done in England, and they were measuring migrating birds and dragonflies that were migrating between England and Africa. They mentioned in the paper that here in North America, it could be even larger. Like the numbers could be even larger in terms of insect migration. So you just have this massive flow. I mean, dragonflies are, bags of phosphorus and nitrogen, right? And they're moving these nutrients from southern.
You're also a bag of phosphorus.
It's
and
true,
nitrogen.
but I'm not migrating like they are in these numbers.
Uh-huh.
But it's just interesting to think of it as this massive flow of nutrients to north and south, back and forth. So that is dragonfly migration.
Very, very cool.
Very, very cool. My mind is just exploding. I wonder if there's historical records of mass seeing masses of
right
before the wetlands were drained and everything.
Yeah, because like you said, places like here, Iroquois, like Montezuma, those places were created because in the 1930s,
Right.
and people started to realize, oh my gosh, we're draining all our wetlands. We're missing, tons of waterfowl.
Yeah,
So-
and the National Wildlife Refuge system is like stopover places.
Right.
For waterfowl mainly was what they were originally established for, but it's good for all kinds of species.
Right. It's not just birds, also
Yes.
insects too.
Just because I'm always thinking about genomes and stuff.
Yeah.
I'm wondering, I'm like, what if the timing of the reproduction is important because the temperature is important for certain gene expression? Because you even have that with sex determination. Sometimes, depending on the species.
And then turtles.
Yeah,
Yeah.
the temperature can be the thing. It's not like you get handed down a Y chromosome or two X's or something. Like, it's just a temperature thing.
So are you wondering if the temperature determines if they're a
So
migrator or
instead
not?
of it being like a sex determining factor, like like with turtles, maybe it's whether you stay or whether you go. What genes get activated to make that decision?
And what activates them, because it wouldn't be just temperature, it could be anything.
Yeah.
Yeah, right. Just
'Cause day
temperature
length is-
or something else. Yeah.
Day length is important for a lot of species in plants and animals.
So it'd be cool to know if anyone's dug into what's the likely environmental factor, or even if it's just a genomic factor. I don't know, but I would imagine it's something environmental.
Cool.
To be pedantic, I should not say day length because the days are always 24 hours. I should say photo period.
There you go.
Right.
All right. Well, to give another shout out to Wild Ideas, we have some stuff for the good of the order. But we're
Oh,
going
yeah.
to do our closing at a later date because right now we're not sure when this is going to come out. So we're not going to do our wrap up right now.
So I should say, Gary, what have you got for the good of the order?
Exactly.
Man, hearing that brought me right back. So good.
So, Gordon, thank you so much for being willing to do this.
No, I was really flattered that you even thought of it.
And I want to give a shout out to the listener who approached us looking for old episodes of Wild Ideas, and that led to this happening. So, us being here at Iroquois with you today. So,
Yeah.
we're so glad it worked out.
And we were clear. I think it's true for Matt too. It's because of Wild Ideas that more or less that we exist and In Defense of Plants exists, right?
Totally.
I mean, we were inspired by that podcast.
Yeah.
So
When you guys all retired, we had a void that we needed to fill.
yeah,
Well, thanks again.
yeah. Thank you, Gordon.
Thank you.
And folks, we'll see you next time. Okay, folks, so we hope you enjoyed that conversation with Gordon. What we're going to do, I'm going to do our normal wrap up right now. Then I'm going to share the episode notes, and then we'll have Violet share our top patrons. So first, for our wrap up, we're going to start with some shout outs. Obviously, we want to thank our listener, Chris S., whose email inspired this episode. And again, we'll keep you updated on where and when you'll be able to listen to all the back episodes of the Wild Ideas podcast. We also want to give a quick shout out to Gordon's Wild Ideas co-hosts, Gary and Joanne, Gary Popotnick and Joanne Ballback. Even though Gordon was the one who joined us for this episode, Wild Ideas was very much a team effort. And Gary and Joanne were all equally a part of what made that show so thoughtful, so curious, and fun. We're grateful for the work all three of them did. We also want to give a shout out to, believe it or not, the New York Times. They recently published an online article called The 7 Podcasts About the Joys of Birdwatching. And folks, it was an honor to be included on the same list as actor Sean Bean, the one who always dies in movies. Apparently he's a big birder and he has a birding podcast himself. So he was number 1, we were number 7 on the bottom of the list. But please don't tell the New York Times that we aren't technically a birding podcast, but we're happy to be included. So thank you to the New York Times for including us there. And we'll share a link to that article in the episode notes. All right, now, we'd like to thank our growing list of Patreon supporters. So thank you to our new patrons, Kristen B., Deborah T., and Rachel C. We also want to give a special shout out each month to our top patrons. So stick around for the end of the episode, and you'll hear my daughter Violet share that list. And if you'd like to support the podcast, consider becoming a patron of our show at patreon.com. Because of support from our listeners, we've been able to keep the show free and make special episodes like this one. Or you can make a one-time donation through PayPal on our website. Remember, you can also help out by sharing the podcast with friends and family and by subscribing and leaving us a review wherever you get your podcasts. It helps spread the word and allows us to reach a wider audience. So come enjoy our sporadic posts on Facebook, Blue Sky, and Instagram, and you can always email us ideas for episode topics, criticisms, or your stories of personal encounters with retired podcast hosts to thefieldguides@gmail.com. All right, so on for the episode notes. Now strap in, folks, because this is probably one of the longest groups of episode notes we've ever had. but they're good ones. All right, during a quick aside this episode, Gordon spotted a great egret, and Steve tried to recall its scientific name, and he tentatively went with Erodia alba. He wasn't entirely wrong. The correct name is Ardea alba, and alba means white, and Ardea means heron, so literally it means white heron. But it also ties back to an ancient myth by everyone's favorite Roman poet, Ovid, and he wrote about a bird rising directly from the ashes of a burned city called Ardea. Now, we also wondered about axolotls. I specifically wondered if they were in the same genus as the Jefferson and blue-spotted salamanders, actually all the salamanders we talked about today, Ambystoma. And yes, they are. The axolotl is Ambystoma mexicanum. And unlike most members of Ambystoma today, which typically metamorphose into terrestrial adults, the axolotl exhibits a trait called neoteny, also called pedomorphosis. This is where an organism retains juvenile or larval traits into adulthood. Remember, if you've seen an axolotl before, they retain those signature feathery external gills into adulthood, and it spends its entire life in the water. All right, now, speaking of Ambystoma salamanders, at one point in the episode, I said that if we found a Jefferson salamander, we would not be able to ID it, just with our eyes. And I wanted to look up if that was true, because I remember hearing something about if it's a male salamander, it would be easier to ID it, more definitively. Now, I would be right that we couldn't identify it if we were talking about a female individual, a blue-spotted or a Jefferson. Many unisexual individuals cannot be confidently identified by sight. That genetic testing was the only thing that would give us a clean answer. But during the breeding season, males do develop a swollen cloaca, and males would not be part of the all-female unisexual lineage. So that can help narrow things down. So a male in breeding condition can sometimes be identified much more confidently than a female, but you would need to have some serious knowledge about species location, morphology, and breeding season characteristics. So you have to be careful. Now, another thing that came up. Going into this episode, I was not aware that the Jefferson salamander and the blue-spotted salamander were part of this larger unisexual polyploid complex. So during this episode, I basically had a, wait, what are we even talking about moment when Steve was explaining the huge all-female unisexual Ambystoma salamander lineage. Steve was talking about a big picture version, this bizarre evolutionary group that can involve genomes from 5 mole salamander species. So remember, those included the blue-spotted, the Jefferson, the small-mouthed, the stream-side, and the eastern tiger salamanders. What I was thinking about was what we hear more about in the Northeast and the Great Lakes region, the Jefferson complex, or some people call the blue-spotted/Jefferson salamander mess. Now, around here, that usually means you can't ID a Jefferson salamander or a blue-spotted down to species because the sale in front of you may belong to the all-female polyploid lineage. So they, as we've already said, they can't be confidently sorted out just by looking at them. And at a certain point in the episode, I asked Steve, if we were in areas where their ranges overlap with some of these other species like the eastern tiger, does that hold true? And the answer is yes, it can happen with the others too. So the narrower Jefferson complex usually refers to the blue-spotted Jefferson part of the story, especially in places where they're the main overlapping species. But when we zoom out and include the other players, again, the small-mouthed, the stream-side, and the eastern tiger salamanders, they usually refer to the whole thing as the unisexual Ambystoma lineage. And the practical field takeaway is this. In places where these species overlap, a female-looking Ambystoma salamander from any of these species may be impossible to identify with confidence by appearance alone. Even experts may only be making an educated guess unless they use genetic testing. So measuring red blood cell size can help estimate the ploidy, basically whether the animal has an extra set of chromosomes, but DNA testing is the real way to know which genomes are actually in there. All right, so when we got to Gordon's stretch of the episode, we were talking about duckweed, and I think I asked, is duckweed the smallest flowering plant? And I believe Gordon said he was fairly sure, but we looked it up, and it is true, but specifically one type of it. We could say one genus of it. The world's smallest flowering plants belong to the genus Wolffia. They're often referred to as rootless duckweed or watermeal, and they are the tiniest members of the duckweed family, Limnaceae. So individual wolfia plants are usually less than 1 millimeter long, roughly the size of a pinhead or a piece of cornmeal, and the flowers are correspondingly microscopic. Now, unlike standard duckweed, which you'd usually find in the genus Lemna, wolfia plants do not have roots, and they appear simply as tiny floating green spheres or oval seeds. And the plant produces the world's smallest flower. It forms in a tiny depression on the plant's top surface. Now, because they're so small and have highly efficient asexual budding, they can easily cover a pond in dense mats before you even notice an individual plant. And they can be found across temperate and tropical regions of North, Central, and South America. So keep an eye out for duckweed, especially the tiniest ones in the genus Wolfia. My next episode note is a correction, and it's about Ed Young's book, An Immense World. During the episode, I mentioned the book and noted that researchers, they're becoming increasingly aware of more and more animals can see in the infrared spectrum, but I was wrong on this. I remembered it incorrectly. It wasn't infrared. What was said in the book is how there's a growing scientific realization that many animals see in the ultraviolet spectrum or the UV spectrum, not infrared. But let's talk about that. Because what prompted me to bring that up is Gordon asking a fantastic question. Can dragonflies see in the infrared spectrum? And when he brought it up, I assumed that he was asking, can they use infrared to help them navigate? So before we answer those questions, first, a quick clarification. When most of us hear infrared, we immediately picture thermal imaging, animals glowing in the dark, predator vision, that whole thing. This is not what we're talking about here. Dragonflies do not appear to see heat signatures. What they may be able to see is what we can call near infrared, light just beyond the deepest red wavelengths visible to us. Near infrared is not heat vision. It's more like an invisible extension of red. So according to a recent study published just this past January, 2026, by researchers in Osaka, Japan, some dragonfly species have visual pigments that are sensitive to extremely long red wavelengths, possibly reaching into the near-infrared edge of the spectrum. That alone is pretty wild, but the discovery also highlights a nice example of parallel evolution between insects and primates. So millions of years ago, our ancestors and other primates evolved molecular changes that helped produce red-sensitive vision, so we could see red. This new research suggests that some dragonflies may have independently evolved a similar molecular tuning mechanism for detecting red light. But while our red vision operates within the visible spectrum, some dragonflies appear to have pushed that sensitivity even farther toward the boundary between, what you can call deep red and near infrared. So what are they doing with this? Probably not navigating migration, at least as far as we know, but they are using it for something. The better supported idea is that this long wavelength vision helps dragonflies identify mates quickly in flight. Male and female dragonflies can reflect red and near infrared light differently, especially against green vegetation. So for an animal making split-second decisions while zipping around at high speed, that extra visual contrast could be a big deal. So to answer Gordon's question, dragonflies may be seeing farther into the red, near-infrared edge of the spectrum than we can see, but based on what researchers currently know, they're probably using that ability for rapid sex recognition, not long-distance navigation. It's more of a high-speed dragonfly dating filter than infrared GPS. All right, so more on dragonflies for our next note. One big piece that I neglected to mention when I was covering how dragonflies navigate during migration is the visual piece, that they also use what they're seeing. And this is a crucial piece of the puzzle. I can't believe I left this out. Dragonflies are extremely visual animals. They have those huge compound eyes, and they're not just decoration. So what they're seeing is likely a key piece in their migration toolbox. Migrating dragonflies are often observed moving along major landscape features. We're talking coastlines, mountain ridges, river valleys, and other long continuous edges. Biologists sometimes refer to these kind of features as leading lines because they can help guide or funnel migrating animals across the landscape. That doesn't mean a dragonfly is looking down and thinking, yeah, that's the Susquehanna River. I'll take the south. But these landscape features, they can still matter in several ways. So for example, coastlines, they can keep migrants from drifting too far over open water, which is risky for an animal like a dragonfly that eventually needs to land, rest, and feed. In mountain ridges and long hillsides, they can create useful air currents and updrafts, which would allow dragonflies, to ride favorable winds and conserve energy. And then you can have river corridors, which provide a visual pathway and also good stopover habitat. They have water, vegetation, plenty of small flying insects to eat, all that stuff that dragonflies need when they stop to refuel. So the overall migration story for dragonflies probably isn't dragonflies have one magic compass. It's more like they're combining a bunch of cues at once. The visual structure of their landscape below them, as well as elements, that we covered during the episode. Okay, one more note here. During the episode, Steve asked 2 really good questions about common green darners. If some are migratory and some are residents, how do they know which group to mate with? And could temperature be the thing that nudges a young green darner down one path or the other to migrate or to stay? So the first answer is actually fairly simple. They probably don't know one from the other, and they probably don't care when it comes who to mate with. For a long time, researchers assumed, I mentioned this in the episode, that groups were reproductively isolated, believing that residents emerged at a different time than the migratory group. But modern research has overturned this idea. According to a comprehensive review, adult flight periods, they absolutely overlap in midsummer. And genetic testing has revealed 0 genetic difference between the 2 groups. The entire continental population of green darners belongs to a single, randomly mating gene pool. So when a common green darner is ready to mate, it's probably not checking whether the other darner in front of them is on the resident or the migratory team. It's just mating with another mature common green darner in the same airspace. Not very romantic, but very dragonfly. Now, Steve's second question gets at something even more interesting. What makes one group migrate while another stays put? This is where temperature, day length, and seasonal timing seem to matter a lot. A green darter nymph developing in warm water during long summer days may be pushed toward faster development. That can produce adults that emerge, feed heavily, build up energy reserves, and migrate south in late summer or fall. But if nymphs are developing as temperatures drop and days get shorter, their development can slow down. Instead of rushing to become adults, they may overwinter as aquatic nymphs and emerge the following year. Those individuals can then become part of the resident breeding population in northern ponds. So Steve's instinct was basically right. Environmental conditions do appear to play a huge role in shaping whether a darner develops quickly and migrates or slows down and overwinters. The only thing to be careful about here is the phrase genetic switch, which we mentioned during the episode. There probably are changes in gene expression involved. Temperature and day length can absolutely affect how insects develop. But in common green darners, we should probably think of it less as a single switch being flipped and more as a whole developmental pathway being shaped by the environment and several cues. So the short version, migratory and resident green darners are not rival dragonfly factions. They appear to be part of one big mixed population whose life cycle can play out in different ways depending on timing, temperature, and local conditions. So folks, those are the episode notes. And here is Violet with this episode's top patrons.
Eric, Alyssa, Adam, Colin G, Mary B, Renz, Abby, Dr. Judy, Kimberly B, Peter R, Callie, The Hebranks, Rich K, Rachel L, The Drunk Phytologist, Orange Julianne, Diane, We Named the Dog Indy, Hot Dog Cold Weather, Dwayne H, Jennifer D, Jonathan A, JDR, Karen E, Deborah B, Are the Plants in the Room with Us Now, Jeremy M, Lori B, Licky Weeks, Virginia A, Catherine W, Holly M, Michaela, Val, John R, Evan, Picasso 3, Mark T, Cecilia, Julie B, Dennis, Chris N, Sung, Jeff S, Jonathan K, Matt E, Plants in My Pants, Ryan R, Sophie S, Connor H, Measure and Principle, Fragaria pepaloinoidea, Outside Chronicles, Quixote, Max D, Melissa Marie, Kelly S, Sarah S, Helen A, Judy F, Ben C, Jane H, Doodle Dude 82, Galen Mac, Kazis, Esther C, John W, Mark V, Bethany H, Rob M.
Thank you for sitting in this hot closet with me.
Oh, you're welcome.
This is our little sound booth in here, folks. So patrons, on behalf of Steve, thank you so much for your support. It helps make episodes like this one possible. And as I mentioned at the top of the episode, folks, if you are a fan of Wild Ideas, stick around for just a moment longer to hear something special at the end. And Violet, thank you.
You're welcome.
All right, see you next time, folks.
Goodbye.
This was fun.
You. Oh yeah, Gary's the one that said it, right?
Yeah, I would always ask at the end.
Yeah.
Since I was sort of the
Right.
host, you know.
Well, can you do? I mean, it would be amazing if you said, "So, Bill, do you have anything for the good of the order?" Because Bill's the one that does it on our podcast.
Could you do it?
Sure.
Yeah.
You're
All right.
Well, Bill, have you got anything for the good of the order?
I do. Parents, get those kids outside. Let them get muddy. Let them get dirty. Let them flip over rocks and logs. And if you don't have kids, get yourself outside.